head the Cancer Biology Institute. And Mark is an expert on receptor tyrosine kinase signaling, and we're going to hear about that today.
Thanks very much. Good. So if I can figure out my presentation. Yikes. So can you hear me okay? Good, thanks. So what I thought I would do today was tell you about a new story that we're playing with in receptor tyrosine kinase signaling, hence the new twists. And basically just kind of start off by running through a few things that we're doing in the lab, just in the sense of, by way of a broad introduction. So receptor tyrosine kinases, which is what we focus on for at least part of the lab, have 58 different members in the human proteome, separated into these 20 different, into these 20 different families. And one of the reasons we're interested in the cancer context, of course, is that all of those which are shaded red here are targets of at least one or other therapeutic, either in the clinic or on their way to the clinic. And as you'll know, in many sequencing analyses, receptor tyrosine kinase mutations crop up an awful lot. So as oncogenic drivers, they're very important. And just looking at them, one sees that they all have an intracellular tyrosine kinase domain. That's what most of the agents target. The extracellular domain is really quite diverse. And that's one of the questions that we've been wondering about over the years in my lab. So the general view, the paradigm, if you like, of receptor tyrosine kinase signaling that all of you know, I'm sure, is that the receptors start as inactive monomers. The ligand has a bivalent character in many cases, and the bivalent ligand binding induces dimerization of the receptor. The tyrosine kinase domains of each of the receptors transphosphorylate one another on tyrosines, leading to the recruitment of downstream signaling molecules that will bind to phosphotyrosines, particularly through SH2 domains, PTB domains, and so on and so forth. So, very simple. And in a sense, I think many people feel that receptor tyrosine kinase signaling is sorted out in a mechanistic sense, but I want to tell you a little bit about that. So I go to meetings about receptor tyrosine kinase signaling in general quite often, and I spend a lot of my time listening to discussions of this part of the signaling network, which is incredibly complicated. And I always feel a bit of an outsider because we work up here, and the receptor tyrosine kinase activation component of these incredibly complex networks. And the systems biologists in particular tend to consider the modules and motifs of signaling in these regions and the network characteristics and so on and so forth. Every single model, it seems, ends up just deciding that the receptor tyrosine kinase activation is essentially a linear relationship between ligand and activation as a simple input into the system, and then all the interesting stuff happens with the downstream network architecture. Here's a simplified version of that. For example, if we look at a few points in a simplified version of this, you can see that we can get, in the case of EGF receptor, a transient activation of phosphorylation of the receptor, which then comes down, phospholipase C gamma downstream of that similarly has transient activation, whereas SHIC at a different point in the network will have a more sustained activation. So different things are happening with different kinetics at different points in this complex network. But it's always assumed that the receptor is just a simple input into that, and the time components of the receptor are not really considered. Complexities and allosteria of the receptor, by and large, aren't really considered. So we've actually always had that in mind and wondered whether we were wasting our time in some senses studying these receptors. But there's an important perspective to think about as you look through now the families of receptor tyrosine kinases and think about that simple dimerization model that we always have in our mind and that sits in the textbooks for the most part. And so just go through a few examples. The TIE is in the middle here, TIE2. We have a paper that will be out in PNAS in a few months, I hope, with TIE2 showing that actually in the absence of ligand, this is a dimer already. So that tells you that receptor has to signal in a different way. We've known that for the insulin receptor for a very long time. So that already tells us that receptor tyrosine kinase activation has to be more complicated and different from from the simple model. Another example, which we're working on quite a lot in the lab, but I won't tell you any of the details of, is a set of receptors, RAW, RIC, which is great for my British R pronunciation difficulty, and PTK7, which is particularly good because it's called Lemmon in Hydra. So that's a receptor I always wanted to work on. These are receptor tyrosine kinases that respond to the Wince, which is a puzzle. And moreover, they respond to the Wince despite the fact that they have dead or pseudo-kinases in their intracellular domains. So these have got to be signaling in a totally different way, which again creates an interesting set of questions for how do receptor tyrosine kinases signal. And not the only pseudo-kinases. ERB3, a member of the EGF receptor family, as well as a couple of the F receptors, and potentially STK106, also dead pseudokinases. So there's a new paradigm that we have to work on there. That's a major area of investigation in the lab. I'm not going to tell you about those things. I just wanted to tell you that by way of suggesting that there's still a lot to learn mechanistically about how these receptors work, which probably explains in part why they're so different in the outside. My initial view of this when I first started working in this field was, this is simple. These receptor tyrosine kinases have a bunch of binding domains in the outside regions. They have a bivalent ligand, and all they need to do is bind to two sites on that bivalent ligand. That bivalent ligand induces dimerization of the receptor. Hey presto, you have a really quite elegant activation mechanism. But it ain't so simple. I think these examples suggest, and so do the preformed dimer examples suggest. So I'm going to tell you a bit about EGF receptor work that we're doing at the moment, which is kind of the nascent work, and we're just starting to write this up at the moment. The EGF receptor was a puzzle back in about 2003. My lab, Kate Ferguson's lab, and several other labs around the world determined crystal structures of parts of this. And I won't go through the details of the structure here, except just to focus your attention on a little bit on the overall architecture of this dimer, but we'll cartoon it and just show this is the kind of paradigm now for how EGF receptor works. But it's too complicated, really, to have the simple view that I was just expounding. So what happens is that in the absence of ligand, the extracellular region of the receptor is in this auto-inhibited state where it has a dimerization arm. You can see there's a little lump there that is actually buried like an ostrich beak being buried under its wing. And that ends up being the primary focus of dimerization in the dimeric receptor. Ligand binds to a region here and here, causes this to stretch open or extend, exposes the dimerization arm, allowing that dimerization to happen. So that seems to be how this works. And then dimerization is induced. You bring the two kinases together. I'm not going to talk much about that, but they allosterically activate one another through a more complicated mechanism than we anticipated. So the puzzle for me here was, why is this so complicated, given that all we need to do is dimerize the receptor? And I think we're starting to understand why. And one of the pieces of data out there in the literature that always made one suspect that there might be something more to this, some more important, more sophisticated, I suppose, allosteric regulation of these receptors, is the fact that if you look at the EGF receptor ligands, there are 7 of them in humans. They come in 2 flavors, green ones and red ones. Green ones bind tightly EGF, TGF alpha, beta-cellulin, and heparin binding epidermal growth factor, like factor. And the red ones are weaker. They bind about 100-fold weaker or so, amphiregulin, epigen, and epiregulin. And what's important is that they signal differently. It's not entirely clear how they signal differently. Mechanistically, it absolutely isn't clear. But what the nature of the differences in signal are is not quite so clear, because the animal studies have been difficult to do. But there's clear evidence that there are differences in signaling. Moreover, if you actually go back, so EGF receptor family has 4 members in humans, ERB1, ERB2, or NU, or HER2, ERB3, and ERB4. In the fly and the worm, we just have 1 copy of this receptor, Drosophila EGF receptor, or LET23 in the case of the worm. But this difference of ligands and high affinity and low affinity ones is also maintained. In flies, we have Spitz, Karen, Gherkin, and Vane. And Vane would be one of the red ligands, and the others would be the green ligands. In worms, you have Lin3. And it comes in multiple splice forms. Some seem to be red and some seem to be green. So this actually goes back the way through when how these receptors are used in development, in patterning during development, which is what they really are important for in the biological context outside of cancer. So there's something fundamental here that's conserved. And we were just wondering, basically, how on earth do these receptors signal differently? One of the hypotheses that had come about as a consequence of our thoughts, and thoughts in particular of David Rees, who was actually a former, he'd worked actually with Dan and also with David Stone here at Yale. This is a review we wrote back together with David quite a long time ago. Was, is it possible that as one sees in G protein coupled receptors, where the concepts of partial agonists and biased agonists are really entrenched in the literature, and we all are happy with that. Receptor tyrosine kinases, if you think about it, everything I've told you so far has kind of assumed an on-off switch. I think that's the way we tend to think about them, partly because of this simple dimerization-based mechanism that has been expounded. Is it possible that for different ligands here, and actually David Rees proposed this for new regulins binding to ERK4, is it possible that different ligands could stabilize different dimers? And as a consequence of these different dimers, you might get different signals that might in some case lead to proliferation, some case lead to invasion, differentiation, or whatever. Is that a possible mechanism of signaling through these receptors, which characteristically only have a single transmembrane alpha helix, whereas of course the GPCRs are 70M receptors, where you can imagine more transmembrane conformational flexibility. So that was really our question. And the answer, which was just came out from work that Dan Fried, who I see is in the audience smirking at me, sorry, and Nick Bestman, over the last year or so, was that actually the dimers with these other ligands are different. So on the left here, and I'll show you a couple of pictures of this, on the left here is a dimer of the extracellular region of the EGF receptor bound to TGF alpha, a canonical ligand. On the right here is the extracellular region of the EGF receptor bound to epiregulin, one of these red ligands. And what you'll notice immediately is there's an asymmetry here. This is a really quite a symmetric complex. This is actually a substantially asymmetric complex. The structure is actually quite different. And if you zoom in a little bit here, you can see the asymmetry here. There's a shift of the right-hand molecule 7 angstroms up compared to the left-hand molecule. Also, the details of the dimerization arm contacts have actually been compromised on the right-hand side here. Moreover, just to give you a sense of the asymmetry, if you look at the ligand environment in the two cases, red as one receptor, the left, and green as the other, you see the environments of the ligand is different. So this is quite asymmetric. Actually reminds us of some studies we published 5 or 6 years ago, where we had illustrated the mechanism of negative cooperativity in this receptor in the fly. So there's some complex allostery going on here. And so we suspected that this might be the reason for the difference in signaling, and we're interested in trying to track that down. As I noticed, the dimerization contacts are impaired there. Because the dimerization contacts are impaired, we wanted to ask, outside of a crystal, would these ectodomain receptor ectodomains actually dimerize? So we used an approach here where we use called small angle X-ray scattering, where we basically zap X-rays at the solution. I won't go through the details, but if you look at the forward scatter, as it's called, which is I0, you can actually get a very accurate and shape-independent measure of molecular weight. If we do that for the human EGF receptor ectodomain and give it an arbitrary molecular weight of 1, with all the controls, of course, add EGF, you can see that we dimerize, go up to 2, and add a little bit more mass because there's some EGF there. We do that with the other ligands that I've mentioned, TGF alpha, HB, EGF, beta-cellulin. They're all quite nice dimers, but epiregulin really doesn't give us much of a dimer at all. In fact, this is pretty much in a monomeric context, suggesting those compromised dimerization interactions may be preventing it from dimerizing, or at least dimerizing in solution. And EpiGen, another one of these, the red ligands, has the same problem. Amphiregulin seems to be intermediate. So we solved then the crystal structure of EpiGen bound to the EGF receptor ectodomain, and it was a monomer in the crystals. We can actually compare the structure of the monomer of EpiGen bound to the EGF receptor ectodomain with a dimerizing ligand bound to the ectodomain, and they're quite different. And one can actually get a sense by looking at the dimerization arm as to why this thing won't dimerize in crystals. If dimerizing this would create all sorts of clashes. So we started to suspect these things don't dimerize at all, and that they may be selective heterodimerizers. So that was our first hypothesis, and I just will tell you immediately that we did a bunch of experiments on that and found that's not the case. To ask whether the dimerization that we see for epiregulin in crystals and the potential dimerization, even with clashes of this ectodomain, is sufficient in cells, we just put these human receptors into Drosophila cells to ask, can they signal? And the answer is, actually, in Drosophila cells, of course, there are no other human EGF receptor family members with which to heterodimerize. So if we see signaling, it must be coming from homodimerization, in principle. And with both EpiGen and epiregulin, we see substantial autophosphorylation of the receptor with the dose-response curve that agrees nicely with their binding affinities. So they certainly seem to work as normal EGF receptors, and if we delete that or mutate that dimerization arm I mentioned, we lose the signaling. So this is by and large signaling that looks a lot like the human EGF receptor, but with compromised or different dimers. So it started to make us suspect one of two things. So they must dimerize in cells. So one of two possibilities, which I'll just give you a little bit of insight into where we are with these. So might these receptors, these ligands, induce this receptor to signal differently because of these different structures? That's one possibility, that where we started. Are they biased agonists? We've certainly seen that the epiregulin-induced dimer is different from the EGF-induced dimer. The EGN-induced dimer, which we couldn't actually see in crystals if we tried to model it, it has to be different. So this situation has to be the case for these two EGF receptor ligands. But the problem with thinking about that in terms of different outputs, as one sees here for the EGF receptor from a simple molecular structure point of view, is that if we look at the EGF receptor, ectodomain, single transmembrane domain, kinase domain, the general view, and our data support it, but we are actively investigating this, is that these red bits have flexible linkages. If that's the case, and this is an issue for every single transmembrane receptor, then it's very hard to understand how we could transmit the difference in detail here to differences in detail here. So we're puzzled by that, and that's something we're actually trying to test to see whether the concept of a biased agonist, if you like, in these dimerizing receptor systems actually holds, can hold water. But the other possibility which we thought about is, might the fact that the receptor dimers that are induced by saturating epigen or epiregulin, the fact they're weaker and more dynamic, because if they're weaker, they're going to dimerize with different lifetimes. Might that be the key, which would actually make epiregulin and epigen not biased agonists, but partial agonists from the point of view of dimerization? And might that be relevant signaling-wise? So we actually did a few experiments here, and I'll just show you some data just to build this up. So we started off collaborating with Dan Leahy and Pat Byrne, where we take the extracellular domain of the receptor plus transmembrane domain plus a fluorescent protein, either YFP or M-cherry, and basically look at when dimers form, you get fret between the two of them. If we do this with the EGF receptor, and we plot the fret against the concentration and density of receptors per micron fluorescence intensity, with EGF, as soon as we can see receptor, we see full fret. It dimerizes very strongly. If we do this with epigen, you can see when we start to get down to lower densities of receptors, i.e. lower concentrations of receptors, you see less fret, less dimer. These dimers are indeed weaker, and the same is true for epigen. The full-length receptor we wanted to study too, which we've done with Diane Lidke, Chris Valley, and Emmanuel I can't remember his last name, sorry, University of New Mexico. And in that context, what we see is that looking at single particle tracking now of the receptor with quantum dots on it in Diane's lab, if we look at a monomer, here's the diffusion, add EGF, here's the diffusion, it's slowed down quite a lot. We add epigen or epiregulin, diffusion is slowed down less, which is consistent with the idea that these dimers are lasting less time before they fall apart, and that's plotted here. But we've also looked, and there are some caveats to this, and this is ongoing, but in the preliminary analysis, the lifetime of the dimer that you induce with EGF is substantially, actually this is the off rate of the dimer, so 1 over the lifetime, but so that basically the dimer that you induce with EGF lasts a lot longer than the dimer that you induce with epiregulin or epigen. So this then starts to make you wonder about the possibility, if this is the case, of something like kinetic proofreading, which in a sense not dissimilar to what one sees in B-cell receptors when kind of distinguishing between ligands of different affinities, which in turn can have substantial signaling consequences. So moving on to signaling with this, we started to look at the timing of signaling. And with EGF, as I showed you at the very beginning, EGF induces a transient activation of the receptor, which then within 10 minutes is down almost back to baseline. But with epiregulin or epigen, which induce these shorter lifetime dimers, the time course is substantially more sustained. So basically the weaker ligands are giving us different and shorter lifetime dimers that are ending up giving us a more sustained signal. Slightly counterintuitive, but what it suggests is potentially that because these ligand-induced dimers are dissociating very rapidly, there's not enough time to assemble the negative feedback groups, which might be internalization, might be dephosphorylation, that are responsible for the diminution of signaling here, which can have profound effects. If you look at this at the level of ERK signaling too, downstream, here you see EGF is very transient, epiregulin and epigen much more sustained ERK signaling too, which I think which was quite surprising to us, and I think is potentially very interesting. And this just reminds one of the story that's been in the literature for 20 years, where if you have a transient signal in many cases, PC12 cells is the classic example, you'll get proliferation. If you have a more sustained signal, it can lead to differentiation. And as you're probably aware, in the PC12 example, there was a lot of work done in the 90s trying to discern what is the basis of the specificity when you have these two growth factors, epidermal growth factor and nerve growth factor, activating receptor tyrosine kinases that seem to use the same set of downstream signaling molecules. How can one lead to cell division and the other lead to neurite outgrowth and growth towards? Where is the specificity? The answer to that, summarized actually one of the best places in this review from Chris Marshall, is that you get sustained ERK signaling in the case of the neurite outgrowth and growth arrest, but transient in the case of epidermal growth factor and its induced proliferation. So this started now to look really quite interesting. And indeed, if we go in MCF7 cells in this case, we use EGF and epiregulin. EGF, which induced the transient activation of the receptor and ERK signaling, gives us nice proliferation actually of the MCF7 cells, whereas epiregulin doesn't, but actually causes enhanced oil red O staining, which is kind of a proxy in a sense, not a very good one, for differentiation in this case. And we're doing some more work to try and understand that. So it really suggests that this small structural differences, potentially these kinetic differences in dimer stability, leading to, we don't know what yet, a loss of assembly of a downstream negative feedback, can actually dramatically change the nature of the response to activating the EGF receptor. And we've done the various controls here to make sure that other receptors that are in this cell aren't influencing this. So basically the bottom line here is that the different EGF receptor ligands do seem to induce different receptor dimers, but with different lifetimes, and we think that might be key. And as a result, different signals. And I'm not quite sure exactly why I put this in, but just an extreme case of this, if we actually, and we've been working on this issue separately, if we look at NGF-induced activation of its receptor in blue and EGF activation of its receptor in red in PC12 cells, there's the dramatic difference between the sustained and transient. And we've been doing a variety of experiments in this context, including inhibiting internalization with Dynamin inhibitors. And by doing that in Dynamin inhibitors, we can change the transient activation of EGF receptor to a sustained activation of EGF receptor in PC12 cells, effectively by switching off the one negative feedback that we could think of. And indeed, in this context, with this inhibitor, you throw EGF onto these cells, you get growth arrest, and they throw out neurites. So really, it does seem by changing the dynamics of the receptor in that way, we can actually achieve these differences between sustained and transient signaling. So I think that was basically what I wanted to point out, just to come back then we've been thinking a lot, and I think the structural revolution in G-protein coupled receptors has really taught us an enormous amount about the details of partial and biased agonism in the G-protein coupled receptor field. And in a sense, the G-protein coupled receptor structures were the beginning of that. So you see them all in Nature and Science every week. Well, not every week, but almost. Whereas in the EGF, in the receptor tyrosine kinase field, one had the sense that the structures were the end, and you can't publish those things in Nature and Science anymore, believe me. And so there was a sense that we've answered the question with the structures here, whereas we began the question with the structures here. And I think what we're seeing is that doesn't look like it's the case. So and actually that receptor tyrosine kinase signaling is a heck of a lot more complicated than we actually imagined, and that partial agonists may be very important here, particularly with respect to the issues of timing. And if you go back to the immunological receptor contexts, there's a great deal of data for the relevance of this, but we've completely ignored it in the receptor tyrosine kinases. So that's something that we're quite interested to think about. And also, when we think about the consequences and the effects of different therapeutics, this is going to have to be brought into line. It offers opportunities. If you can bias the receptor in one way or another, of course, there's one aspect. But it also will reveal some limitations in some of the drugs that we're currently using. So I think as we delve through this, there are going to be some important and interesting things to look at. So I will stop there and just thank the people who've done this work. And basically everything that I described here was really done by Nick Bestman, who was a graduate student who really initiated the project. He was a graduate student at UPenn before we came here, and is now working at Weill Cornell as a postdoc. Dan Fried has taken over the mantle and has pushed this forward and has solved the epiregular structure and done nearly all of the structural analysis that I just mentioned. Dan Fried came with us to Yale from Penn and is an associate research scientist here. And Anatoly Kyatkin has been working closely with Dan on the cellular aspects of this, and Anatoly is a research scientist. So I will, I think, stop at that point and hopefully